My paper is distinct E-cadherin-based complexes regulate cell behavior through microRNA processing, or SERP, and P120 cantamine activity, or in very simple terms, restoring proper function of cancer cells. So special thanks to D, Ms. Legerifo, Sydney Stein, Mark Strockman, the Journal Club team, my mom, and my family. So, quick story about me getting into the research team. So, at first, when I came to Queen Greek, didn't like science, never came to Journal Club, and then I had Mr. D as my Bio 1 teacher, and I kind of liked it, so he convinced me to go to Journal Club, kind of liked it, and I wanted to take the cancer class, but in the end, I couldn't take it, and I was really determined because I really wanted to take a class about cancer, so I ended up taking a summer course about it and doing all these things, so in the end I just really was too determined to let anything get in my way. So here I am now. So now, what is cancer? Can anybody name some characteristics of cancer? Talking specifically to people who have taken the cancer class.
Unrestricted cell proliferation.
That's good. That's one of them. Anyone know anything else?
Angiogenesis.
Yeah.
Inhibition of apoptosis.
Good job. Anyone else? Okay. Well, those are my three main points. So, accelerated proliferation, that's uncontrolled cell growth and division. The second one is angiogenesis. A lot of people don't know what that is, but angiogenesis is the recruitment of blood cells. And so the reason that cancer cells do this is so that they're able to nourish themselves to continue this accelerated proliferation. The next is resisting apoptosis. Apoptosis is cell suicide, so essentially they don't die when they should. The next two are really important to this paper. So the fourth here is invasion. And so, say we're talking about melanoma skin cancer, invasion is when the cells are able to go through multiple layers, essentially invading the tissue around it. And then metastasis is very similar, so it's gone so far invading that it goes into the bloodstream, and it'll travel through the bloodstream to a different spot, and will create a cell colony somewhere else and create a tumor in a completely different spot. And so both of these have to do with motility or movement of the cells. And I want you to keep motility in mind. So the current therapies right now are surgery, chemotherapy, and radiation. Can anyone tell me what these all have in common? Yeah.
They're destroying or removing the cancer cells.
Very good. So they're all focused on killing these cells. The problem with this is when you can't specifically target normal cells from cancer cells because if you think about it, they're still your cells. So it's very hard to only kill the cancer cells and not just regular cells. So that means that it becomes highly toxic when you're killing both. And it's often very invasive and intrusive, but also you can become resistant to current treatments. So now we're going to dive into the paper. So what they knew already, okay, so they looked into epithelial cells. So epithelial cells are formed in layers. So here we're talking about E-cadherins, so that's two epithelial cells. They're epithelial cadherins, if you can hear it, adhere, so they make them stick together. And P120 stabilizes this adhesion. So without P120, these adherents are able to essentially be absorbed into the cell. And so without those two things, the cells essentially are able to move and become motile, which is big in cancer, so you don't want that. And that's why P120 and E-cadherins are often known as tumor suppressors. Yes. Yes. So, two epithelial cells next to each other. So they're often linked by E-cadherins. And so without E-cadherins, they would be able to move. So P120 stabilizes these E-cadherins because without them, the E-cadherins would be able to be essentially absorbed into the cell. So that means that with P120 and EK present, it makes the two essentially tumor suppressors. And the problem is P120 is also known to be an oncogene or pro-tumorigenic, but we don't know why, and that's a big contradiction in previous knowledge. So now we're going to get into the paper. So, do you want to, yeah?
Are they tumor suppressors because they suppress metastasis by keeping the cell fully together, or for different reasons?
Also invasion, because when they can't move, they can't do those last two things. Invasion and metastasis. Do you want to turn off the lights? I think that'd be easier to see what's going on. Okay, so what they first started off doing is, so there's two parts of the epithelial cells that they focus on: the apical, the top, the basolateral, the base, and the sides. So they wanted to isolate where P120 is found. So they know that pleca was only found apically, or on the top, and P120 is found on the, and we see here that P120 is found on the base and the sides. So this on the right is a silver stain. It's essentially like a Western blot. And the green means apical, and the red means basolaterally. So you can see pleca is only found apically, while P120 is found both basolaterally and apically on the cell. Yeah?
Can you just clarify what pleca is? I'm sorry.
It's it'll make more sense later. But you're gonna see, here, okay. So this will answer your question. So we know what P120 does basolaterally. It stabilizes these coherence, but we want to know what it does apically and how that has to do with pleca, because we don't really understand yet.
Are you just asking if that's a protein?
Yeah.
No, I just don't understand how it did.
No.
So we're gonna build. Okay, so what does P120 and pleca do together? That's what we are going to build. So here you see what pleca does, what effects it has on the cell, which should answer your question. So here on the right we have a immunofluorescent staining of pleca and actin in cells. So this is a pleca knockdown. Pretty much shRNA just means that they're knocking down pleca here. So can anyone tell me, based on these two or these images, what you think pleca has on the cells? What effect? Pretty much comparing the top to the bottom too. No? Okay. Well, as you can see here, the cell structure isn't as intense. And there's, as you can see, obviously less pleca because it's being knocked down. So saying fold change is proliferation and cell growth, and this is the control, and these are the pleca knockdowns. What other effects does it have on the cell based on C? I'm calling you out.
Me?
Yeah.
You don't want my answer.
This is proliferation, or growth. So the bigger bar means.
More growth.
Yeah, so without pleca, cells are able to divide more quickly. So here you can see with P120 in the apical, with pleca being knocked down, there's less ability, less structure, more cell growth, and more colony formation. Bad things, unhappy target. So we want to see, okay, what does P120 and pleca both do apically, and how do they affect the cell? So if we're looking on the left here, these are the knockdowns. So these are what they're using to knock pleca down in P120. So the negative means that it's the control group not using those knockdowns. The plus means that they are using the knockdowns, and the double plus means they're using both knockdowns. So fold change is the same. It means proliferation. What do you think happens here when, what do you think that this is trying to tell us? Yes. Okay, well, when both of them are at a normal rate of normal expression, they're being normally expressed, there's a normal amount of growth. But when, as we saw earlier, yeah?
What determines like normal expression?
I don't really know how to answer that. It's just,
I think she was like, how do you read it?
how do you rate normal?
Like, are those percentages relevant to what it would normally do?
Yeah, so this is without it being knocked down, so that's would be the control for normal. And so they're saying in the second one, with only pleca knockdown, as we saw before, that it's increased, the fold change is increased. But with the double knockdown, we see that it's been lowered again. So essentially, the division has been lowered again of the cells. So why are we talking about growth? So here, these are all growth factors. So this is a Western blot, so these are, as you can see in the middle, when only pleca is expressed, down-regulated, there's more expression of these growth factors. So that means downstream, something is happening when pleca is decreased to make these growth factors increased. So why not stop here? Could anybody tell us, okay, so we figured out that when we when we knock down P120 and pleca together, we lower this proliferation rate. Can anybody tell us why we wouldn't stop here? What's the importance of P120, why we can't knock it down? Yeah.
It keeps the cells from moving.
Yeah, exactly. So we can't just stop here because when we knock down P120, it's going to stop those adherents from sticking together. So we have to keep looking, okay, what else can we do? So now we know there's growth factors downstream of pleca, and without pleca, it can't be inhibited. But what's this last question mark? What's missing? So we knew that pleca had something to do with the microRNA processing. So microRNA, it's essentially another system to regulate expression of proteins. And so they did this whole screening of when pleca is knocked down, what else is affected, and they noticed that microRNA 30B was the most drastically changed out of all of them. It was extremely decreased. So they wanted to see, okay, what happens if we can put those microRNA back in? So here you can see this is the control without the mimic microRNA. This is the control when they added the microRNA. This is the pleca when they didn't add microRNA, which is what we've been seeing, how there's an increase in those same growth factors with when pleca is decreased. So we're essentially seeing the same thing here, but what's the difference between the third and the fourth when they add the microRNA back in, these being growth factors? So there's less expression of these growth factors when microRNA 30B is put back in, when they re-express microRNA 30B. So essentially what they're telling us is that downstream of pleca, there are microRNA that control these these growth factors. So basolaterally, P120 acts as an adhesion stabilizer, but apically, the reason that it can be an oncogene we figured out is that it'll keep expressing these growth factors. So pretty much what we figured out that when pleca and P120 are expressed evenly, those microRNA are able to suppress those growth factors. So what does this mean? Pretty much as I just stated, we clarified the roles of E-cadherin and P120 and pleca all in cancer. So can anyone remind me of the two oncogenic and the tumor suppressor roles of P120.
Wait, say your question again?
Can anyone tell me the two different roles of P120?
Cell adhesion.
Cell adhesion, and what's the other one? To make it oncogenic or tumor promoting.
It's like increase the growth factor.
Exactly. So now we figured out that, so now we know the role of E-coherent in P120, and pleca helps suppress those growth factors with P120. So now we've also figured out that mimicking these microRNA, we're being able to rehabilitate the cancer cells. So what does it mean? So we've now found a potential therapy that needs to be tested in vivo. And we also have to find additional less destructive treatments. So that has to do with the rehab versus destroy therapies. So as I stated earlier, the current treatments of surgery, chemotherapy, radiation all have to do with killing these cells. What if instead, as was just shown, we can rehabilitate them and not have to kill everything, making it less toxic, hopefully. Anybody have any questions? Yeah.
Can you just go back, excuse me, to the finish, the pathway?
Yeah.
Finalized?
This one or the other one?
This
Oh, well, the other one.
So, is the top here represents the tumor suppressing, the bottom represents the oncogenic, or is it more complicated?
It's more complicated. So P120 basolaterally with the growth with P120, it also has to do with that cell-to-cell adhesion. So basolaterally, it can be tumor suppressing, but also P120 without pleca, that's what makes P120 oncogenic.
It doesn't.
Because it doesn't create those microRNA to suppress those growth factors. Anybody have any questions? Yeah.
So does this only target cancer cells? Because if you're suppressing the growth factors for healthy cells that are essential to create, making essential proteins, then how is it only suppressing the cancer?
They didn't say in the paper, but can I make a mention? Yes. So if you
So as long as you're making it clear, that's
it's an educated guess. I think it's because what they almost need like when pleca is only downregulated, that's when there's actually, you can see that in normal cells here, when they added those microRNA 30B, there's really no essential changes of those growth factors. So my educated guess is that it wouldn't affect them significantly. Any other questions? Yeah?
Do you think this approach of using microRNAs will be useful in regulating a lot of different oncogenes, or is it specific to this system?
Well, microRNA are very widespread and unknown, I'd say. So they control a lot of gene regulation. So I think that they could use it for a lot of things, but I don't necessarily know what their next step is specifically with different